also online. And I just start over again. So what I was just saying is I'm very happy to be here, first of all. And also thanks for the really nice organization here on location. It's been a pleasure so far. I'm going to pick up a bit where Silke left it off yesterday because she already gave a really nice introduction to chytrids, and I'm also going to be talking a lot about chytrids because that's first of all that's the main group of organisms I work with, and also the examples that Serena will be working later will also be on chytrids, right, Serena? I hope so.
I confirm, yes.
Perfect. So I also switched around a little bit my presentation because Silke already told a lot of things yesterday that I also wanted to say, so that was a really good introduction. So it's not super polished, but let's see how far we get. I can just use this one. Sorry. Where do I click? Oh, just. First of all, parasites. I just wanted to give you a couple of numbers because I think these are actually useful numbers and also interesting numbers to put things into relation. And one of these numbers that really kind of struck me already in my PhD time is that actually 40% of all species in the kingdom Animalia are parasitic. You see this really nice paper by Andy Dobson, where he just looks at 25 or more different taxon groups. And then on the y-axis, there's the proportion of parasitic species in that taxon group. You see the usual suspects, of course, but 40%, I thought this is a lot. Another paper that really impressed me was a study looking at the diversity. I'm actually in the middle of the picture for you guys. Let's go here then. This is weird, sitting, presenting. Yeah. So this paper I thought is really nice because it looked at over 200 lakes in the Pyrenees, across the Pyrenees. It used meta-barcoding to look at all the different genus and species groups they could find in these different lakes. And they actually found a pretty nice relationship between producers, so mostly the phytoplankton. And then if you can see it, these reddish triangles, that's putative parasites. And actually then my favorite group, of course, the chytrids, they're right up here. So they're both species rich and also very abundant per lake. And they're also contributing a lot to the species richness in these lakes. So parasites are not only many, but they're also very diverse. And they're also kind of often a bit hidden. That there was also a study I really liked by Lefebvre, looking at all these heterotrophic flagellates and then trying to put a number to how many of these are probably parasites. And with her approach, she also showed that a good third of these heterotrophic flagellates might be parasites. Also a bit of a newer study by Comeau, looking at different habitats, seawater, freshwater, but also ice, which I think is very cool. He looked at the fungal community there, and everything in green is chytrid or chytrid-like. So me very happy, favorite group, strongly represented. So there's a huge diversity, and it's also to a large part not very well described. So that also gives people like Silke and me a chance to continue this job. And here you have a couple of pictures alongside of phytoplankton parasitized by chytrids. They're all from Lake Marseille in the Netherlands and Rübelsee in Germany. Good morning, everybody. Right. So they're also diverse. So what do they do in the food web? And that's a pretty cool study by Claire Gachon, where she looked at the food web, and on the basis you have the phytoplankton, then in the middle you have consumers and zooplankton, and then in the top layer you have fish. So that's a typical food web like you would expect to see everywhere. But of course, the parasites are missing. That's what Claire added to this picture. So if you add the parasites to the picture, that is actually only the parasites of the phytoplankton. Yeah, true. You see that you get a lot more connections. Again, you get 40% more species. You get four times the number of trophic links. So the linkage is changing. The whole structure of the food web is changing if you include parasites in your calculations. And it also doubles the connectance. So these food web links, they become much tighter. So that also says something about where the nutrients and the energy flow is going in these systems that we often do not, we do not include. So, do they also have biomass? And that's another one of my favorite studies by Amor Kouris. And he looked at 3 estuaries in California. And what you see here is, of course, not very readable for you guys, but basically they calculated the biomass of all the free living species going from small clams and things like that up to the overwintering birds. So that's the overwintering bird biomass here. And then they went one step further and they calculated all the parasite biomass, which is in this system a lot of worms actually. So you get all these biomasses, then they grouped it, and then what you basically see is here's again the biomass of the overwintering birds on the y-axis. These parasites have a lot of biomass. Actually, they also compared it to putting 3.5 elephants on these little estuaries. And if you put the elephants there, nobody would say like, yeah, they're just there, they're not doing anything. So this biomass also must have influence on the nutrient and energy cycles. Morning. Right, so we now showed that biomass, they make connections, they have diversity, and they're actually pretty much everywhere. And a nice concept to kind of bring this all together was always the disease triangle to me, which is very simple. Basically, you have 3 components. You have the parasite and the host, let's see, and then the environment which they're exposed to. Obviously, the environment influences the host. We all know that. It influences also the parasite. We're getting there. There is an interaction between the host and the parasite, independent of the environment. But I think the most interesting part of that concept really is that there is also this arrow going from the environment onto the interaction. So the interaction is modulated by the environment these parasites are. This host parasite system is living in. And then environment doesn't only have to be the physical environment, but it can also be the community that's where it's all taking place. And so think about grazers. We had a couple of questions about grazers yesterday. Think about competitors. Yeah, diversity patterns. Right. Quick recap in case you forgot what chytrids are. You will never forget again. So they are a very early diverging fungal lineage. Actually, there are microfossils of chytrids. This is an example from the Rhine chert, a specimen by the University of Münster, 300 million years ago. And this is actually an infected phytoplankton with a sporangium, chytrid sporangium, they think. So it's a very conserved association, actually. There's more than 750 species described and counting. They do occur in actually absolutely every aquatic environment, also in inundated soils, in just the little water layers on top of everything. So wherever you look, you will find them. And they have a very diverse set of consumer strategies, going from saprotrophic to parasitic, to even hyperparasitic. And that also means that they have a very wide range of substrates, but also a very wide range of specificity. And there's really cool work by Silke van Wijngaard and Michael Kagami trying to chart out how general or how specific these chytrids can be. And that ranges from chytrids that can infect several species across taxonomic groups to chytrids that really actually only do well on very few genotypes of one species. So it's a full spectrum. They have this really nice life cycle where they have both asexual life cycles and sexual life cycles. The asexual is more common, which basically goes from these free-swimming zoospores that go out, search a host, and attach to it, invade it, form this sporangium outside of the host, in which by mitosis they form new zoospores. These are released and then the cycle repeats. But every now and then they also go through a sexual cycle where you kind of get this, this conjugation tube going on, and a resting spore is formed. And these resting spores, they can, well, to my knowledge, we don't have any clue how to wake them up. So they stick around for a long time. At some point they will hatch again, and then the cycle starts again. But that also shows that there is potential for evolutionary trajectories, depending on which life cycle is going on. And together with that, the generation time of the host is kind of similar to the generation time of the parasite in this case, which is different from other parasitological systems. I mean, if you think about worms infecting a giraffe, the giraffe has a much longer life cycle, right? But here it's a matter of days. They have the same speed, so they have the same potential to co-evolve, and that gives different opportunities in terms of co-evolution. Yeah. So these chytrids, they of course occur in an environment, and one of the environments that I've looked at in a lot of detail, and also Silke looked at in a lot of detail, is Lake Maarsseveen in the Netherlands, where Ellen van Donk actually started with monitoring the lake in the late 70s. Did she? No. Do I have it now? Yeah, indeed. And here in these little graphs, if you can see them, yeah. You have in the blue line is actually the temperature development in the lake. Then you have in black the host and in red the parasite. And what you see is that, yes, you do get relatively constantly a spring bloom of Osteonella in this case, but some years also not. And then it is or isn't followed by a epidemic of the parasite, which is relatively specialized to this host. And then the question was, why do we get this different patterns in different years? And we kind of zoomed in on temperature in this case, and on host density. And what came out of that study basically is, yes, the parasite is has a host density dependence. If there's more host, then it does better. Not too surprising. And there is also a little bit of a threshold below which it doesn't work that much, that it doesn't transmit that well anymore. But what really came out is that the winter temperature, a couple of weeks before the epidemic starts, is the most decisive factor. And that it was a little bit of a paradox, because actually in cold winters, when the parasite has to go into when the parasite has to go into a resting stage, we got strong epidemics. And the mechanism that it works is, in cold winters, the host can already start growing. It builds up biomass. When it warms up, the parasite wakes up, it becomes active, it has a lot of biomass to prey on, and it makes an epidemic. Whereas in warmer winters above 3 degrees, the parasite stayed active. It constantly preyed a little bit on the host, and both of them didn't do very well. So in that case, actually, with a warm winter, you lose the infections. Then we were interested in my PhD how that affects the genetic diversity in the host population, because also Osteonella is mostly asexually reproducing parasite diatom. And we thought, we would actually see a very uniform host population with only a few genotypes, but that was not true. Actually, you see quite a bit of variability, but it depends on whether you have an epidemic or not. So my first year of a PhD, obviously, of course, no epidemic and no bloom happens. So if you want to get rid of your parasite, just put a PhD and get them started on it. Everything disappears. But we went out, we took samples. Luckily, the next year, the next 2 years were better again, and we got this kind of nice and the parasite epidemic following it. And we tried to kind of sample at the start of the bloom and at the height of the epidemic for the host population. We used AFLP, which is a not very much used genetic barcoding method these days. But then at that time, it was a good method. And then we looked at the diversity, the genetic diversity. So in the year with no bloom, the diversity was relatively low, and it also stayed low. In the year with a bloom and parasite epidemic on top of it, we actually got this increase in diversity. And the idea here is that the parasite can adapt to the most common genotype. It will prey on that genotype, and that gives room for the other genotypes that were less competitive to come up and become more numerous. And so that is one of the examples how the parasite can affect the evolution of a host locally. Then another system I looked at in more detail is Müggelsee, close to Berlin. It's a shallow lake as well. Has a quite a strong agriculturally dominated catchment, so it's also a very eutrophic lake. And the special thing about Müggelsee is also there since the 1980s, late 70s. There's a constant monitoring system going on. They go out, take phytoplankton samples every week. And they have also all the physical and chemical data. It's a really neat data set. But the cool thing they also did is that they have all these samples that they maintain fixed in formaldehyde. So I could actually use those ones and go back and start counting the chytrid time series based on these samples. They were in formaldehyde, so no chance of doing anything molecular. So I went to the light microscope, and we just used a really simple method of making like a blood smear, just a drop, add a stain, in my case, calcofluor white. But looking at the success that Silke has with the wheat agglutinin, I think next time I want to try that one. And just count it really. And what came out of that is a time series I'm pretty happy with. Here you see in black the total phytoplankton biomasses, and then in red the infected phytoplankton biomasses. And what you see is several years. You can see the years by the water temperature in light blue. And what you do see is, yes, there are blooms, and they are also followed by infections. But the picture is much more messy, right? It's the infections are there the whole year. There's not really a break in winter. It goes down, but it doesn't completely disappear. You see sometimes a lot of biomass with no infection. So what's going on here? But what you do see, and I thought that's also pretty cool, is that like almost 75% of the time points that were looked at, we found infections. And the average infection prevalence was 3%, which you would not think is a very much, but if you just think like constantly you remove 3% from all the populations, that is actually quite a hit on the phytoplankton. We found that 17 host species of phytoplankton taxa were recurrently infected, and that occurred in all groups, so in cyanobacteria and diatoms in green algae. And what you see here on the first bar in this graph is actually the proportion of samples in which the host occurred, and then the second bar is the proportion when the host occurred that it was infected. And you see a little bit that sometimes if something occurs more often, it also is more often infected. But you also get the other picture that some species that hardly ever occur are quite often infected, or they occur more often, but they're hardly infected. So you get all kind of combinations. Obviously, not everything is equally well infected, and not all the time. And this graph here is just the overall biomasses. So, of course, the phytoplankton has the highest biomass in the system, followed by the herbivorous zooplankton. And then the carnivorous zooplankton actually has the same biomass as the chytrid infected hosts. But that's what it tells you is that also in this system, these tiny things do build up quite a bit of biomass. Just to show you a few examples, so Alocosira was one of the most constant bloomers, but also most constantly infected species in the lake. You see quite typical patterns of summer blooms followed by epidemics. Arsenella had much less biomass in the system, but also was constantly infected as soon as it popped up. But then you get cases like Plantotrix, which builds up quite some peaks of biomass. So this is 10 milligrams per liter. Sometimes it gets infected, but other blooms are completely uninfected. And one explanation for that could be that these cyanobacteria are known to produce oligopeptides that also inhibit the proteases of chytrids, and that could also actually stop the attachment of the parasite. And then you get some examples where a species rarely occurs and it immediately, as soon as it pops up, it gets infected. And that's also the question, where does this parasite stay in the meantime? Is it on other species? Is it just waiting somewhere as a resting spore? Because these are long stretches, right? There's a couple of years in between these skeletonema occurrences.
Where is skeletonema in the meantime?
Yeah, also a question. I mean, maybe the very easiest answer would be that it's somewhere in the catchment. There's some reservoirs up there. But so far, we're not entirely sure. Could also just be on the sediment, or below detection limit, which is, with my counting method, is not all that low. So we started to figure out a few things about this system, and one thing we were interested in is, of course, and that's also the topic of today, is what is driving these infections. And the first thing we tried is to look at regression trees to see where does it split. And for most of the systems that we looked at, the first split is really at host biomass. So the host has to be there, makes sense. Otherwise, there's no infection. But then in the third or fourth tier of these regression trees, the environmentals pop up. So especially nutrients and temperature. So if the temperature is above a certain threshold or below a certain threshold, you do or do not get infections. Another thing that we could now do with this data set is that we looked at how are these chytrids embedded in the food web. And for that, I made groups of chytrids here in yellow that are linked to their specific host group, so the diatoms, edible and inedible diatoms, and edible, inedible green algae, edible for the zooplankton and others. In blue, these are omnivorous or herbivorous zooplankton groups, so the rotifers, some cladostra, some copepods, and mussel larvae, actually. And in purple here are the carnivorous zooplankton groups. And then we built an autoregressive model to kind of arrive at the weighted and directed network. And what you see here is in green are positive and in red are negative interactions with an arrow. So for example, one of the strongest interactions is a positive interaction from this phytoplankton group to their specific chytrid group. What is interesting here is to see that, so now this is the same network, but then I took everything away that had no connections with the chytrids. Obviously, the phytoplankton groups positively influence the chytrids that are specialized to them. But also you do see some red interactions between the herbivorous zooplankton and the chytrids. And that means that if you have at one time step more zooplankton, the next time step you will have less infections. So they seem to probably eat some of the zoospores and then reduce the infection load. What is also interesting is here that some of the strongest links are from the phytoplankton to the chytrid, not so much from the phytoplankton to the herbivorous zooplankton. So then that raises the questions about, yeah, what happens with the competition between phytoplankton in the presence or absence of the parasite? And I was able to use systems isolated by Silke. I'm really happy, and these are absolutely fantastic photos that she's taken of her system. So check out these papers, it's really cool. So we did an experiment where we really looked at if we have two host species competing and we put a chytrid specific to the winner, what happens then to the loser? We used Synedra and Starostrum, or now yeah, these days, and Starostrum as a model system, and we worked with these kind of fed batch cultures. So I would every second day I would take out 30% of the culture and replete the medium to keep the system going and to have the hosts compete for the nutrients. We had per treatment we had 10 bottles. The treatments were either replete, so high phosphorus, high silicon. One was low silicate to exclude or to drive down the diatom, and one was low phosphorus to drive down the starostrum. And this is what happens if you don't put any parasites. If silicon phosphorus are replete, the green algae will win. If silicon is low, green algae will win again. If phosphorus is low, then the diatom will win. And then at the red time step, when actually was clear who was winning the competition long term, to 5 bottles we added the parasite specific to the winner. And you see this here in these kind of orange, that's the parasite epidemics. And you see that the picture really turns around. The loser is now winning. In 2 cases, the winner was completely eradicated from the cultures. And here, actually, we got into a new equilibrium with very low Synedra, very low infections, and a bit higher starostrum. And that also made me question, because we always think that if parasites kind of control the winners, then the losers can come up, we get high diversity, but that's not necessarily true for every single system. And these are very simple systems. So for these simple systems, that doesn't hold. Then we're also interested to see what are the grazers doing. So we added, we made a food chain from Synedra, which is not edible to rotifers. We put parasites on it and the rotifers on top of it. The black line is the zoospore numbers in the control treatment, so no grazers. And then the blue lines are all different amounts of grazers. And you see the grazers do graze away quite a bit of the zoospores. But it does not change the outcome of the infection. The infection still goes up to almost 90% in this case, even with the highest amount of grazers. And that was really because the grazers could only graze down the zoospores to a certain level, because below that level, they started dying off themselves. It was not enough food for them to maintain themselves in the absence of other food. And with Takeshi Miki and Maiko, we then arrived at this model where we kind of looked at where is the balance, how much grazers can reduce the amount of zoospores in a system in the absence of other food, and how does that relate to their body size. So we took a lot of literature values, we took our own study, and Takeshi made this magic and put it all in a model. And basically what you have here is on the y-axis is the body length of the grazer, and the clearance rate by the grazer, and then the lines here are actually the equilibrium lines down to which level of prevalence of infection the grazers can reduce the infection. And you see some of, if it's up on these lines, they don't really do a lot. But if it's down here, they can actually reduce infection a lot. And that is especially true for the smaller zooplankton. So the ciliates, but also here, it's not written, but really at the lower end here would be the amoebae, for example. So there's a clear relation on who can really reduce the infections. So bringing that whole picture together, we kind of figured out that organisms, and well, obviously it's well known that organisms live in an environment and they react to that environment and they have a tolerance range. And often the tolerance range is kind of hump-shaped. And that is true for the host species, but also for the parasite species. And if they have exactly the same tolerance range and the same shape, you would basically say, yeah, wherever the host is, it can be parasitized. But that's probably not true all the time. And there's one hypothesis, which is called the thermal mismatch hypothesis by Cohen and Jason Frureau. And they hypothesize that parasites are usually smaller than the host, so they have faster generation times, they have faster adaptation capacity, so they should be able to live in a broader range than the host, at higher temperatures, at lower temperatures than the host. Which really only makes sense if the parasite can somehow endure this time where there is no host, either as a resting stage or on a different substrate. But the opposite picture is also observed. So the parasite has a narrower range than the host. And then you get this interesting situation where you have environmental conditions that are not optimal for the host, but there's also no disease. So that might actually be better than the optimal condition where there is disease. And of course, since we're ecologists, we also wanted to know what happens if we put global change on top of that. So we handily excluded evolution, and we wanted to kind of look at what happens if these environmental conditions disappear. The cold winters disappear. This disease refuge is then also not available anymore. But also maybe summers become hotter, and then you arrive at a new disease refuge. And we do have empirical examples both from the field and from the lab for this. This is also one study of my PhD, where you have on the y-axis the net production of infected cells from over the time period of the experiment. So if that's close to zero, then nothing is happening. There's no infection. If it's higher, then you have higher infection across different temperatures. What you see here is, in the case of Acinella deals with 1 degree and 21 degree beautifully. It's not even close to their limit. But the parasite can't. So at 1 degree, it doesn't do much because it's actually forming resting stages. So it doesn't really disappear from the system, but it's diluted out and it's not active. At 21 degrees, it just dies. And the Acinella is still kind of hanging on there for another 5 degrees or so. So there is actually a winter refuge, and there's also a summer refuge from disease. Another example is for salinity, and I mean, several of you mentioned salinity yesterday as well as one of the tricks and tips to deal with some of the parasites that you encounter. And this is a study by Kindles, I guess, where she exposed Planktotrix and their parasite, their chytrid, to a range of conductivity conditions. And you see this, this really steep drop to a low infectivity, but then, so this is the percent increase in infected filaments again, down to actually a negative infectivity at the highest salinity or conductivity range. And in their system, it's not so much about sodium chlorides or something like that, but it's much more about nutrient salts that are washed into the lake by rainfall and a lot of agriculture around. So that was then also kind of topped up with another experiment they did on turbulence and mixing. So these zoospores, they actively swim to find a host. There is most likely chemotaxis involved, so they have to have some sort of there can be water movement, but if there's too much water movement, it's quite likely that first of all they can't swim so well, and second of all their gradient is disappearing. And that's basically what you see here, with increasing mixing or turbulence in the water. These parasites don't infect so well anymore. So that's another potential refuge from disease for the host. And a bit more of a subtle refuge is the case of where the ratio between nutrients makes a difference on whether the parasite can infect well or not. Here, Thijs looked at the ratio between phosphorus and nitrogen, and from earlier studies, we always assumed that the parasite, the chytrids, really need high phosphorus. They need to build a lot of DNA, they have to build a lot of proteins, they need a lot of RNA, so that they should need a lot of phosphorus. But actually, our experiment here showed that they actually really want to have nitrate and nitrogen. So in blue, you have the growth rate of the host, which is relatively constant across a range of N2P ratios. So a low N2P ratio actually means less N, and a high N2P ratio means more N relative to P. And then here in orange, you see the parasite, which is doing worse at low N and better at high N conditions. So this is probably not a completely exclusive disease refuge, but it is with modulating the amount of, or the ratio of different nutrients, you can modulate whether or not you have a lot of infection. And that is probably also true for micronutrients, and that is also an area that has not been explored at all, how dependent these parasites are on micronutrients. A little bit more far-fetched, but this is an example of also changes in disease dynamics under pollutants. In this case, this was an experiment with diclofenac, and diclofenac is an anti-inflammatory drug that is used very widely. It's also not fully metabolized, so it ends up in a lot of surface waters. And we wanted to test how that affects the host parasite interaction, because there's actually very little ecotox work on, apart from fungicides, for example. But even those studies are rare. So there's very little ecotox work on host parasite interactions in the aquatic and in the fungal, the aquatic fungal world. So what you see here is a gradient of diclofenac concentrations increasing. In this kind of mid-blue line is actually the host if it's not infected. The dark blue line is the host in the infected treatments, and this blue line with the what's it now? Yeah, light blue is then the infection prevalence of the parasite. And you see at low diclofenac concentrations, not a whole lot is happening. The parasite is happily infecting, but at some point you get a switch And actually the parasite is not dealing so very well with the drug anymore, whereas the host is not all that much concerned. It's not increasing as steeply anymore as before, but still doing quite well under the diclofenac concentrations. And then in the end, you get a very subtle scenario at the highest diclofenac where actually the host is in the end doing not fantastic, but still better than the parasite. So also in that respect, pollutants can also shift again, or can also be a driver of infection success in these systems. And of course, these are all very nice and simple examples. Most of them are only one environmental variable, and that's not exactly what's happening out there. So really one of the open questions is how does this all work if it's several confounding, interacting environmental variables? Do we then get at these multivariate pockets of disease-free zones, or does it actually just not matter so much at all? Some of our field examples say yes, strong drivers can matter because, like, for example, in Lake Marseille, but also in Norway with Plantotrix examples, we could see that there are temperature refuges for the host. But are these the exceptions, or is this the normal thing that happens out in nature? We don't really know. So that calls for more work, and it also is, of course, then the question: is this ecological knowledge something that we can use to apply it in the industry, in water management also? And how would we do that? And that actually links then a little bit to what we want to try this afternoon. So this afternoon, 7.Alena is having an open workshop for statistical problems. And I wish I could just add on and get myself advised. But the other part is that we will just design studies. So I will ask you to form groups, to formulate your own hypothesis, which can be something that you're struggling with in real life, or if you're parasite free in your life, then you can just come up with your own question. I just want to work with you guys then through the process of having formulated a hypothesis, thinking about ways to test it or to work with it, and then come up with an experimental design or something else that we can then present to the whole group again. Yeah, by that, I'm going to finish, shut up, and answer questions.
Thank you, Alena.
Just for time.
Yeah. Thank you very much. So, any questions for Alena also? Maybe Maya?
Yeah. I would just like to say that it's amazing what you presented us here today. I think we have a lot to think about, but I see that we will have to do research on every system. Everything is possible, but what troubles me, let's say, the most is this. You answered my question. If I have a culture and it doesn't work so well, probably there is some parasite that coexists and they are both in trouble, not doing very well. So then it's a question: it's better that I have continuous culture and it is alive but not doing well, or should I have this batch cultures with a burst of biomass production and just trying to harvest it before the parasites takes over or something like this?
Yeah.
A lot of playground here.
Yeah, and I mean that's a super difficult decision, because both have costs.
Yeah.
And you don't really know the long-term risk of, you might in the end get some sort of co-evolution in your system if you have a long-term, if you keep it running with a low infectious load. which can be beneficial if the host decides to become resistant by miracle, but it might also work out the other way, right? One thing I'm always a bit wondering about is, of course, these chytrids are also interesting in terms of their nutritious properties.
Would be the best.
That would also be interesting to see whether that can be an avenue, but that's also not true for all the parasites, I guess. So, yeah.
No, but the idea that at the end you would have algae as a food for the chytrids and then harvest chytrids and have products is also an idea.
It could be, yeah, it could be. Getting it stable is the next problematic issue. Yeah.
And it would be really great if there was such a research also on our ponds or not, like.
Well, we have a lot of SCSMs here, and that could actually come with new things. So there's opportunities within the cost, and we're still going strong for another 2.5 years.
Yeah.
So, and I mean, the ambition after this network is really to move on to a bigger European network with actual money for research. So that's yeah. So it's good to think about already, like where are like the nukes where we can hang the hat to make that happen. Yeah. Cool. Online, everybody happy? No questions.
No
question.
Good.
Maybe we should check in chat.
Chat, no questions about.
No questions.
Yeah.
Okay, thank you,
Thanks.
Alena. Thank you.